All right, now we're ready for the next step in protein synthesis, and that is translation. We just did transcription, which was making an RNA from a DNA. Now let's translate that into a protein. And so you remember that making RNA from DNA is called transcription. They're in the same kind of language. Now we're going to go into a different kind of language, so we need to translate it. So the messenger RNA has been processed, it leaves the nucleus, it's ready to be translated. What organelle do we need? Of course we need a ribosome. And you remember there are free ribosomes floating around in the cytoplasm, and bound ribosomes, which are attached to the endoplasmic reticulum to make rough endoplasmic reticulum. So the ribosomes are going to read that piece of mRNA and make a polypeptide. And as I mentioned before, the ribosomes are made of a type of RNA called rRNA, ribosomal RNA. There's also a little protein here. It's a big organelle. The scaffolding is made out of RNA. And there's a large subunit and a small subunit. And those two subunits are separate until they're ready to translate something, then they come together. You recall these are synthesized in the nucleolus, inside the nucleus, then exported, and that's when they come together to translate the mRNA. And so here's a figure from your book, and you see the two subunits that come together to make the ribosome. And what they're showing in this picture is accurate, that you can have several ribosomes working on the same RNA. And so, you know, that RNA is carrying genetic instruction, right? It's like the master copy, but then you can read it multiple times and make a new protein every time. And that's what they're showing here. You can make a bunch of polypeptides all at once, and it's very efficient. So the ribosome binds to that mRNA, then it reads the mRNA, and that tells you what amino acids to put in what order. So it's just like we talked about. Earlier we had polymerase reading the DNA and bringing in nucleotides, or for either DNA or for RNA. Now we've got the ribosome reading the RNA and bringing in the proper amino acid in the proper order. Now those amino acids are carried to the ribosome by another kind of RNA called transfer RNA, or tRNA. So the tRNA has got an amino acid bound to it. And so that tRNA comes in and is also bound to the ribosome. It's got hold of the mRNA. Now you bring in a piece of tRNA, but that tRNA is bringing an amino acid with it. And so this new amino acid is added to the end of the growing polypeptide, and then you shift over and you just do it again. And so you read the mRNA piece by piece. That tells you what amino acid to bring in. You just stick that, so it's just like when we grew DNA or when we grew RNA. We grew it from one end. We're doing the same thing with the polypeptide. Once that tRNA has given up its amino acid, then it gets released. And that tRNA is going to go and eventually find another amino acid and do it again. But while that's happening, the next tRNA with amino acid comes in, and then it's released. Then the next one comes in and it's released. And so just amino acid by amino acid, you build that polypeptide. You keep doing that until you got the whole polypeptide or the whole protein. So the ribosome is reading the mRNA, and that's determining the primary structure of the polypeptide. You remember, primary structure is the order in which the amino acids go, and that determines everything else. Primary structure determines secondary structure, determines tertiary structure, determines quaternary structure. Change that primary structure and you might change the protein. But what determined this mRNA sequence? Well, that came from the DNA, from the gene that was just transcribed. And so that is how DNA makes RNA, makes protein. So ultimately, the DNA is sitting up here controlling everything, but you see now how it controls what proteins are built. Okay, so let's get to a little bit more detail about this. As that ribosome reads the mRNA, and it tells it, which amino acid to bring in next. Well, how? How does the ribosome know which amino acid to use? How is it able to, take what's on that RNA and turn that information, translate that information into an amino acid? So how can we translate this piece of mRNA? Remember, we're taking one language and converting it to another language. So we're going from the nucleic acid language into the amino acid language. So we need to take words, again, quote unquote, from the mRNA and translate them into amino acids for a polypeptide. Well, in this particular metaphor, all the words only have 3 letters. And so obviously, the English language, we've got all these words, and they have different lengths. In this language, all words are 3 letters, and those 3 letters are called a codon. And so when the ribosome reads the mRNA, it reads it codon by codon. Before, with polymerase, we went base by base, right? And then that you brought in the new nucleotide based, from one base, you knew what the proper base was to bring in for the growing RNA or the growing DNA. Here we got to read 3 nucleotides at a time. We got to read a codon, and the codon is what tells you what amino acid to bring in. So there's your translation. So then the tRNA brings in that proper amino acid. The ribosome catalyzes all these reactions. Remember, this is, RNA and protein. It's a big kind of, almost like an enzyme. So it's going to take this new amino acid, add it to the end of the polypeptide that's been growing, and then shift over to the next codon and get the next amino acid, and then shift over to the next codon. So we read the mRNA 3 bases at a time, or a codon at a time. And so in this example, you see the first codon codes for tryptophan. The next codon codes for phenylalanine, the next codon for glycine, and the last codon for serine. So we've translated from nucleic acid into amino acid, into a polypeptide. Well, again, how do I know this? How do I know, for example, you see U, that codes for phenylalanine. How do I know that? How does a ribosome know that? What's interesting is that those codons always only mean one amino acid. And so, for example, GCG, GGC, which you see in this example, is always glycine. No matter what the organism, if a ribosome reads GGC, it's going to bring in a glycine. And so that's pretty cool. And so because of that, we can come up with a chart like this, which is what's known as the universal genetic code. And this is all the different possible codon combinations that you would find in an mRNA. And for each codon, it tells you what amino acid. And this doesn't change no matter what organism you're in. And so if you know the codon, you know the amino acid. It's just like earlier, if you knew the nucleotide, if you were doing making DNA or making RNA, if you knew one strand, you knew the other strand. Well, here, if you know the codon, you know the amino acid. So since you've got 4 bases in RNA, taken 3 at a time, there's 64 possible codons, and they're all represented here. But there's only 20 amino acids. You remember from the protein chapter that we talked about that there's only 20 amino acids that are used to make your protein. So each amino acid has several codons that code for it. It's redundant. You also see in this chart there's a couple of codons that are start and a couple of there's one start and there's a couple of stop codons, and those are also give information to the ribosome. But what's really fascinating is that this is the same for every living organism. And so I'm a fish guy. I didn't have to learn a special alphabet or a special code for fish. And if you were, if you're a fern person, you wouldn't have to learn a special code. It's the same for every living thing. That's really cool. So if a ribosome in one of your cells is reading mRNA and it reads AAA, then it's going to stick a lysine in to whatever polypeptide is being grown. And if a ribosome in a cactus is reading an RNA and it reads AAA, it's also going to bring a lysine in. That's the universal genetic code. Okay, so we mentioned there's a start codon. So that's what the ribosome looks for. It looks for the start codon, and it starts building the polypeptide there, one amino acid at a time. And so you read the codons one at a time. Each one represents a single amino acid that gets added to the growing polypeptide. And then you eventually hit a stop codon. The ribosome lets go. Now you've got a polypeptide. And that polypeptide can go off and, maybe it'll go to the Golgi apparatus and get modified a little, or maybe it'll go do its thing, but that's it. So that's how we're translating the mRNA into a new language. We're building a string of amino acids based upon the information in the mRNA. And so again, we emphasize that DNA is just a long string of nucleotides. There's no branches, it's one long string. And RNA is one long string. And proteins are one long string of amino acids. Now you understand why they are, because they all come from each other. It all works together. And so here's just kind of an overall diagram, just giving you a feel for this. You've got all these tRNAs floating around and they have an amino acid. You've got the ribosome, you see the large subunit, the small subunit. You see it's reading the codons, 3 letters at a time. And you see the tRNAs are bringing in the amino acid. You see the growing polypeptide. All the stuff we just talked about. So here, same thing, we just zoomed in a little bit more. And you can see that you've got basically 3 sites for a tRNA. You've got this E site, which I'm not sure, maybe it's the eject site, I'm sure. But the E site is where the tRNA that no longer has an amino acid gets ejected. You've got the P site, that's the middle one. That's where the growing polypeptide is sitting. And then you got the A site, the active site. That's where the new tRNA comes in. And you see that last one has got a single amino acid, but that amino acid is going to get stuck to the end of that growing polypeptide. And so, in a little bit more detail, again, we say the ribosome reads the codon, but how do you get that correct amino acid? Well, it's actually the tRNA that helps with that. So the tRNA comes in, and it has an anticodon. And so it's just like we did with DNA and with RNA. If you know the nucleotide sequence, you know what will pair with it. And so if you know the codon, you know the anticodon. And so there's only one tRNA that will fit each anticodon. And each tRNA carries only the amino acid that's appropriate for its anticodon. So these tRNAs are not just grabbing any amino acid. Whatever the anticodon is on that tRNA, that determines what amino acid it gets. And so now that anticodon can pair up with the codon, and that's how you bring the correct amino acid to the growing polypeptide. So there you go. And so this is what a tRNA looks like. It's not a very long molecule. And you see up at the top, that's where the amino acid attaches. So that's how it carries the amino acid. And then directly opposite that, you see the anticodon. And it's got this, you've got these funky little curves, and it's along that curve where you've got the anticodon. And so that anticodon can only pair with a certain codon. And so this anticodon will only pair with a UUC codon. And that's the only time that this anticodon will be allowed to come into the ribosome. But this tRNA will always have a phenylalanine. It will never have any other kind of amino acid. And so the ribosome reads a UUC on the RNA. So I want to make sure that we're clear. Whenever I show you one of these charts, again, look, it says, first mRNA basic. These are always based on the mRNA. These are always based on the codon. So the ribosome reads a UUC codon. The only tRNA that it will allow to come in is one with the proper anticodon. This one with the AAG anticodon. But you can see from this chart that tRNA should be bringing in a phenylalanine. PHE is phenylalanine. And so that's what we're saying here, is that when the ribosome reads that AAG on the mRNA, this is the only tRNA that can come in, and this tRNA will be carrying a phenylalanine. And how do I know that? Because I know what the codon is, the UUC codon is only for phenylalanine. So what you got to do is you got to just think about all this and realize, and just make sure you understand the terms. And so you know who we're talking about. Where do you find the anticodon? Where do you find the codon? The chart is based on codons. Keep it all straight in your head. Okay, here's a figure from your book, kind of just showing you the same thing. Showing an anticodon and a codon matching up. So here is this diagram again, and you can see the tRNAs and the codons and the anticodons and how they all relate to one another. So again, we can look at the steps here. Here you can see the ribosome is holding on to a tRNA in the P site, and that's where the growing polypeptide is. You see that polypeptide growing off the top of it? So the A site, the active site, is where the new tRNA comes in. And so, only the tRNA with the proper anticodon can come into this active site. And so once that happens, what they're showing you in this step is that polypeptide being transferred over to the new tRNA and the new amino acid. Do you see that little red arrow there? And so this is how we add the new amino acid to the end of the polypeptide. We're actually adding the whole polypeptide to the new amino acid. Now you also notice a little thing there that says GTP. And GTP gets turned into a GDP and a phosphate, right? Does that look familiar? It should. It looks very much like when we talked about ATP earlier. And it's very much the same thing. You remember ATP is a nucleotide with 3 phosphates. And whenever we need energy, we remove one of those phosphates, and there's a ton of energy in that bond, and we use that energy to do something. Well, there are other nucleotides that have the same structure, and here instead of an ATP, we've got a GTP. It's a guanine triphosphate. I'm not sure exactly how you pronounce it, but it's it's just the same nucleotide that we've been using, but instead of having 1 phosphorus, it's got 3 phosphoruses. They all can do that. It's just ATP is the one that's used all over for all the cells all the time. But every once in a while, you'll see this, GTP, carrying in oxygen, or carrying in energy. And so that energy is used to shift the growing polypeptide to this new amino acid. And so in this stage now, you see that the polypeptide is on the new tRNA. The old one doesn't hold anything. Now the ribosome goes, it clicks over to the next codon, and so the old tRNA is now in that E site, and so it gets ejected. The new tRNA is now in the P site, and it's got the whole polypeptide. And now we're ready to read the next codon. And so we're ready to bring in the next tRNA, and so on and so on. So you can see how this cycle is just, read it, bring in the tRNA, transfer the polypeptide, read it, bring in the tRNA, transfer the polypeptide. That's what the ribosome does to build the polypeptide. And so here they're just kind of showing you the same thing again. And so now we've got all the steps diagrammed here. The DNA that gets transcribed into the RNA that gets translated into the polypeptide. Okay, so now we can take a lot, we can look at several different possible things that could happen. Let's say that the gene has a sequence ACG on the template strand. Remember, the template strand is what gets read to make RNA. So what would the RNA sequence be? Well, if the DNA is ACG, the RNA must be UGC. Right? We don't use thymines, we pair a U with the A since we're making an RNA. So that's going to be the mRNA sequence. So if the mRNA codon is UGC, what amino acid should go into the polypeptide? Well, you can't memorize, don't memorize this table, right? That's a waste of time. If I ever ask you any of these questions, I'll give you this table. And so you got to look up UGC in this table. And now if you look at the table, you can see that on the left you've got the first code, so you find U. On the top, the columns, you got the second code, and so you look for G, and then the third gives you the third codon. And so that's, you can find your codons that way, or you can just kind of look around, however works for you. But if you look at UGC, you see it up there in the upper right. It's a cysteine. And so from that DNA, we got the codon. From the codon, we got a cysteine. Okay, so let's pretend, same gene, but it has a mutation, right? The sequence changes. So that ACG gets changed to an ACA. There is a mutation. What's the new RNA? Change DNA, change the RNA, right? The new RNA must be what? UGU, right? And so if that's the new codon, if the mRNA codon is UGU, what's the proper amino acid? So we go back to our chart, we look up UGU, oh, and we see it's the same. It's a cysteine. So the DNA changed, and that caused the RNA to change, but the protein did not change. You're going to put the same amino acid in this location. And so that is a type of silent mutation. The DNA mutated, but it did not change what amino acid goes in this position, so the protein is exactly the same. And so that's, that's why we say not all mutations are harmful. I've been saying all along that you change the DNA, you definitely will change the RNA, and that might change the protein. Now you see why I say might. The DNA changed, the RNA changed, but you got the exact same amino acid in the exact same spot, so there's no way of telling. You got the exact same protein. Silent mutation. All right, let's do another mutation. Let's say that the original ACG mutates to ACC. What's the mRNA from this? Well, if you're transcribing it, it should be UGG, right? And so if the DNA is reading ACC, the RNA should be UGG. And so if that's our new mRNA codon, what's the amino acid? So let's go up and look up UGG. UGG. Oh, that's a tryptophan. Uh-oh. So in this example, the DNA mutated, which of course changed the RNA. That has to happen. But in this example, you did get a new amino acid in that position. And so you will get a new protein. Now, this protein might still behave like the original. It might be that that amino acid in that position is not supercritical. And so if you swap out an amino acid, the protein still works fine. So that would be another type of silent mutation. The DNA mutated, you put a new amino acid in, but it ended up not making a difference. That can happen. It's very possible that that new amino acid is going to mess everything up. Remember, proteins are all about their shape. That primary structure is all, determines all the secondary, tertiary, quaternary structure. Shape is everything to proteins. Changing the primary structure often will radically change everything about that protein. And so it's very likely that if you change an amino acid, the protein is going to be worse off. But every once in a while, you change that amino acid and the protein's a little bit better, and it works a little bit better. It's a little bit better for the organism. And so mutations can do nothing. They can be deleterious or harmful, or they can be beneficial. It all just depends. Well, that's it. You now know the steps for DNA to RNA to protein. You can call yourself a biologist because this is like the most important idea in biology. Okay, so the last thing I want to talk about is the universal genetic code. I've already sort of mentioned this, and we've seen the table. And so gene expression and what genes do and how they're expressed, that's all different among all different organisms. But the idea of a gene is universal. We all do this DNA to RNA to protein in the same way. It means different things in different organisms, and we do it in different ways, but the basic idea is the same by all living things. Okay, and you learn right away in biology, you can't say, you can't make broad statements like that. There's always going to be an exception. And yeah, there's little exceptions, just little ones here and there. But as a whole, the smallest bacteria, the tallest redwood, all living things and all living things that have ever existed all share this genetic code. And that's pretty cool. So an AAA codon means a lysine in a human or a cactus or an E. coli. That's pretty cool. Well, what that means is you can take a gene from one species, put it in another species, and that gene will keep on trucking. It will be transcribed and translated as if it was in the original species. And this is how we can make transgenic organisms. We can take genes from one organism and put it in another, and it'll still work because the code is the same. And so I can take a gene from one species, put it into a different species, and it will make RNA and then make protein in the new species. That's cool. This works because DNA to RNA to protein works the same in every living thing. And the code is the same in every living thing. Now the take-home message here, or the corollary to this idea, is that this is pretty strong evidence that we all came from the same common ancestor. This is why all living things are related. If you go back far enough in evolutionary time, we all have the same ancestor. Because if you have very broad things that all living things share, well, where do we get them from? Where did you get it from? You got it from your parents. Where'd they get it from? From their parents. Where'd they get it from? Their parents. And you keep tracing that back, we get all our traits from the generation before, the generation, our parental generation, and they got their traits from the parental generation. And so if you have this trait, it had to all have come from an early ancestor. Now, is it really that simple? Can you just take a gene and just throw it in another species? It kind of is. It kind of is, and then it kind of ain't. The idea is that simple. It just works, no matter what organism. The practical aspect is what's complicated, right? And there's all kinds of genetic control. And one gene, a lot of times depends on lots of, there's lots of other reasons why, that make this hard to do. I can't just go to a lab and grab it. But it's also, once you do get it figured out, it does work that simple. And so it's not something that you can learn to do overnight, but it's something that we can do very easily. And it's pretty cool. And it's been done in a lot of ways, in a lot of cool ways, right? And so if you look here on the left, you've got a tobacco plant. And this tobacco plant has had a gene from a firefly spliced into it. And so you take the gene from the firefly that makes luciferase, which is an enzyme that when it cuts molecules apart, that it gives off light, and that's how fireflies are able to light up. They use this luciferase gene. Well, you take that gene, put it into a plant, and that plant will transcribe and translate the protein, and that plant will light up. Or here you got a pig with a jellyfish gene, and so you got another gene that's in jellyfish that makes them luminesce. And so you take that gene, put it in a pig, and then in certain places that gene will get expressed. So if you look at this pig, you can see it's not the whole pig's not glowing, the snout and the hooves are glowing. And so what that means is you probably put that jellyfish gene right next to like a gene that makes keratin, right? Because keratin's that tough protein that you find in hooves, and a pig's snout's going to be tough. And so you probably got that gene right next to that keratin gene. So whenever the keratin gene got turned on, so did the jellyfish gene, and it made them color up like that. Pretty cool. Okay, so our next step is we're going to practice doing this transcription and translation. And we'll practice this a lot, and I'll ask you a lot of questions about this. It's very important. So you ask me questions if you have them. Otherwise, it's getting dark, so I better go in. See ya.